In the last discussion, we tried to provide you with some background, some of the processes that are going on to create differences between sequences, and then some of the things that can complicate our measurement and our analyses of those sequences. So to help us bridge the gap between observed distances and actual distances, we need to implement models of evolution that allow us to correct. Now, realize that once we reach that saturation point, that models of evolution are really no longer useful because there's just too many mutations that have occurred and we can't correct for all of them. But when we're kind of in that gray area where we get the observed distances diverging away from the actual distances, the model of evolution is really, really critically important. And so it's useful for a wide array of different things. Now, when we do models of evolution, we can simply, I guess maybe not simply, and I know this is complicated and I know we're kind of jumping right in there, but if you are struggling a little bit, if you're not totally getting it all, you can go back over it in the lecture if you want to, but maybe more effectively is if you log on to one of the Zoom sessions or email me and make an appointment, ask questions, ask for us to do more explanations and go over it again. So I'm here and I'm available if you're not doing it. So when we are looking at models of evolution, we need to estimate the probability of different types of changes. So we have a little matrix here, all of the different probabilities. There are 16 possible changes that can occur. Now, the P here represents the probability of a mutation, and then these little letters represent the type of mutation that is occurring. So this is the probability of a C mutating into an A, the probability of a G mutating into an A, the probability of a T mutating into A. And although we have 16 possible mutations, in reality, we disregard 4 of them because this is not really a meaningful thing. The probability of an A mutating into an A, an adenine into an adenine, that's really not meaningful. So in reality, we only have 12 mutations that we need to really assign values to. And these values can be anywhere from a 0, meaning no possibility at all, to a 1, meaning 100, which is not really realistic because in real situations, you never have 100% chance of all A's changing into a C or C's into an A. So these are going to be somewhere between 0 and 1 and usually fairly low. Okay, but we could assign values to those. Now, how we get those values, we'll get to at a later point. Okay, but just know this is kind of a theoretical framework for assigning a probability of a mutation. In addition to that, if we're going to do a model of evolution, we need to know not only what the probability of a certain type of mutation is, but what are the frequency of the 4 different nucleotides in the sequences that we're looking at. What's the frequency of adenines in it, the frequencies of cytosines, guanines, and thymines. So this is simply frequency of the different base pairs or the different nucleotides. So relative frequency of bases, or if you want to say nucleotides instead of bases, that's fine. There are different terms for this, and I know it can be confusing, but essentially nucleotide base pairs or bases and nucleic acid sequences, those are all the same. Okay, so just be aware of that. Okay, so we've got these 2 different parameters, and we are going to implement models of evolution that are of varying levels of complexity based on these probabilities of mutations and these frequencies of the different base pairs. So the first model that we're going to look at is the simplest of all models. It's called Jukes and Cantor. Many of these models are named after the people that first described them in the literature 20, 30 years ago. Okay, so unfortunately, they're not very descriptive names, but you just have to remember them. Well, actually, because your quizzes and exams are online, you don't have to remember them as much for this class, but you need to be able to go back and put the correct model of evolution with its name. So Jukes and Cantor has 2 basic assumptions, and this is the simplest model of evolution. We are assuming that the frequency of all of the base pairs is equal. They're all going to be 1/4 or 0.25, 25%. Okay, so that there's equal number of cytosines, adenines, guanines, and thymines in the DNA base pair. That's a very simple assumption. Okay. The second assumption is that there's an equal cost for all of the different types of mutations. Here we've represented it with the Greek letter alpha, but notice that I think that this was an A to a C. The probability of an A changing into a C is the same as an A changing into a T as an A changing into a G. right? So we assign equal probabilities to all of the mutations and equal frequencies of base pairs. That's Jukes and Cantor. Now, whether or not this is biologically realistic is another question. So this may or may not be the best model to fit what we're actually seeing in the sequences. And sometimes a simple mutation or simple model is not the best one. So we have more complex models. Model number 2 that you need to know about is called Kimura two-parameter. And again, named after the person who first described it. This one's a little bit more descriptive if you want to remember it this way, because there are 2 parameters that we're estimating when we're looking at the mutation rate or the substitution rate. Okay, so we're still going to assume 25% or 0.25 for all the frequency of base pairs, but we're going to have a difference between the probability of a transition versus a transversion. So here, if we go back to our original matrix, C to A, G to A, T to A. So a G to A, which is a transition, we assign 1 probability, represented here by the Greek letter beta. A transversion, we assign a different one. So transitions and transversions get different probabilities of occurring. And this model was originally developed because when we looked at real DNA sequences, often we observed that the differences that could be categorized as transitions were much, much higher. There were many more transitions than there were transversions. And so we needed to give them different probabilities. That is the Kimura two-parameter. Felsenstein, 1981, again, this is not very a descriptive name, you just have to remember it. In addition to having, Felsenstein is changing the frequencies of the base pairs, but having the exact same probability of a mutation. And so now we're going to combine the frequencies of mutations and add them in with the probability of different mutations. So the frequencies, I misspoke. The frequencies of the nucleotides, and then add them in with the probability of mutations. So we've got different possible frequencies here represented by this little pi A, pi C. These can all be different numbers now rather than all being the same. But notice that the alpha here is the same for each of the possible mutations. Okay, so simplistically, the assumptions for Felsenstein 81 is that we have different frequencies of the nucleotides, right? But an equal cost for all types of mutations. The next one, the next logical extension, is called HKY85. That's what we'll do. That's the easiest one, described by these 3 Japanese researchers. And now we combine, if you notice, this HKY85 is basically a combination of the Kimura two-parameter and the Felsenstein 1981, where we can have different frequencies of base pairs and a different cost for transitions versus transversions. Okay, so each one of these as we move up is getting a little bit more complex. Jukes and Cantor is the simplest. Kimura two-parameter is 1 level more complex because of this. This is also 1 level more complex. We left the mutation probability the same, but we changed frequencies of base pairs, so that's 1 step more complex than the Jukes and Cantor. And by the time we get to this one, now we are 2 steps more complex. This is a more complex model of evolution than the Jukes and Cantor by 2, because not only do we have to estimate different nucleotide frequencies, we also have to estimate different probabilities for transitions versus transversions. And then finally, the 5th model of evolution that you need to know is called general time reversible. And in this one, we have much more complexity here, because not only do we need to estimate different frequencies for the different base pair, the different nucleotides, we also need to estimate a completely different rate of mutation for every single possible mutation. So the reason this is general time reversible, I misspoke there. We don't need to do it for every single mutation, because a C changing to an A is estimated the same as an A changing into a C. We don't make a distinction between the direction of the mutation. But notice an A to a C, a G to an A, whatever. I don't have this matrix memorized, so that may not. In fact, let's go back and look and see exactly what it is. So this is an A to a C and a C to an A. So notice here that we have an A to a C or to C to an A. Both that probability of mutation is represented by the letter A, and it's the same no matter what direction it occurs. And then B, and then here we have the one G and C. So each of these little italics represents a different type of mutation. Okay, so this is the most complex type, the most complex model of evolution. And here are the assumptions. Again, normally I have my students memorize these 5 models of evolution. Familiarize yourself with them. You maybe don't need to memorize them because on quizzes and exams you could refer back to this. It might be handy for you to make notes. And in fact, writing it down in and of itself will be helpful. But make a note or write down these 5 different models of evolution and how they treat these different assumptions. Now, in addition to these different assumptions, there are 2 other parameters that we need to add to models of evolution. And these are added on to whatever base model we're going to use, whether it's the Jukes and Cantor model or the HKY model or the general time reversible model. We can add on one additional parameter. One of these is the distribution of rates. And when I say rates, I mean rates of evolution. And what scientists noticed is that depending on the DNA sequence, some areas of that DNA seem to have a much higher substitution rate, seem to evolve and mutate faster than other areas of that DNA. And so they realized that we might want to incorporate that into our model of evolution, that there might be parts of the DNA that mutate faster than others. Just to give you an example, let's go back to our original DNA sequence that we were looking at. So I'm going to open it back up. I closed down that program. And you'll get more and more familiar with this as you move on. Let's see if I can find the one align. Open a saved alignment session. Let's see which ones we have. Sorry. I think it's right here. Open a recently used file. That's the one I want. So this is the one we were looking at earlier. Okay, so here is our DNA sequence. And we're just going to look at the end of it here where it's much fewer insertions and deletions. And what we're going to do is we are going to compare the mutations that occur at the 3rd position versus the mutations that occur at the 1st and 2nd position. And if you've had an evolution class, hopefully this is somewhat familiar to you. But you'll notice a pattern up here where we have everything's the same, and look, then there are mutations, 2 mutations. Same mutation. Same mutation. Same mutation. Is it getting redundant here? So those of you who have had my evolution class, or perhaps someone else's, that's a familiar pattern to you. And the reason that we see a much higher rate of mutation at this position is because it's the 3rd position in the codon. And if you don't know why that is, don't worry about it. But realize that we might want a different model of evolution for 3rd positions than we have for 1st and 2nd positions because there's just higher substitution rate there. So that's just one example. And we're not going to get into the complexities of this. This is as detailed as we're going to get, which is probably much more detailed than many of you really would like to do, but you need to know it, okay? So the gamma distribution is simply a statistical method for estimating how different the rates of substitution are at different sites. And so we could add on, in fact, let's go back and let's type it in here. Oops, I'm going to go back in here. Sorry. There we go. So we might have, for instance, a very simple model like Jukes and Cantor model. And then in addition to that, we add in a gamma distribution. Now, if rates of evolution are the same at all sites, then we don't need to include a gamma distribution. But very, very frequently, those rates of evolution at different parts of the gene or at 3rd position versus 1st position are different. So we do need to add a gamma distribution. We can also, right, if we had a more complex one, like the most complex one we learned, the GTR, we could also add in a gamma distribution. Okay, so realize that our models of evolution are getting more and more complex as we add in these additional parameters. So in the Jukes and Cantor, we have 3 parameters. Well, I'm sorry. If we did Jukes and Cantor plus gamma distribution, we would need to estimate 3 parameters. We need to estimate a substitution rate, which in Jukes and Cantor is the same for all. I'm going to make this a little bit smaller. So for this one, we would need to estimate 3 parameters. We would need to estimate that substitution rate, which is in Jukes and Cantor assumed to be the same for all mutations. So that would be alpha, and we would need to estimate that based on our data. We would also need to estimate the, or put in our assumption of 25% frequency for all base pairs. And then in addition, we would need to estimate the gamma distribution. So that's 3 parameters that we would need to estimate. For this GTR, we would actually need to estimate the 6 different parameters for the different base pairs. So if you go back to this general time release, we would need to estimate A, B, C, D, E, and F. So that would be 6 different parameters we would need to estimate. Plus, we would need to estimate different rates of evolution here. So that's at least 3, maybe depending on how you cut it, 4 more. So that's 10 different parameters. Plus, then we would need to estimate the gamma distribution. So for this one, we need to estimate at least 10 and maybe as many as 11, depending on how you cut it. If that doesn't make sense, that's fine. That's not important, the number. But we would need to estimate many more parameters for this model of evolution. Okay? So we'll get to this in a little bit. How do we pick which one we're going to use? Do we pick the simpler one where we only have to estimate 3 parameters, or do we pick the more complex one where we have to estimate 10 parameters? We'll get to that in a minute. Before we do, we need to go to one last parameter that is often used, and this is called the proportion of invariable sites. It's estimated by, or it's represented by this letter I. So what we may need to do, and basically what this means is, how many columns in the alignment do we have that have no changes at all? What is the proportion of invariable sites? And it might be very, very high in some sequences where we have very few changes, and very, very low in other ones. So the proportion of invariable sites becomes a final parameter that we are going to learn about that we need to estimate. So we might now, and I'll just do it back here. We might want to add a final parameter to our model of evolution where we would add in a proportion of invariable sites. And so we would estimate 4 parameters now. Here we'd now be up to as many as 11 parameters if we added in our proportion of invariable sites. Okay? So, for your purposes, I know this is complex. I know some of you are like, your head's hurting a little bit. Sorry, I apologize. Some areas of this class are going to be kind of complex, technical like this. Others are going to be a little bit more accessible. Hopefully you followed along with most of this. If not, review it, log on to a Zoom session, talk to me about it, ask questions about it where you're still confused, okay? But now we've set up this idea where we could estimate models of evolution, simple, going all the way to more complex. We'll talk about how we pick which of those models is the best here in just a little bit. But for your purposes, review, make sure you are familiar with the 5 basic models of evolution. Realize that we can add on to those models of evolution by adding a proportion of invariable sites, or by adding a gamma distribution, which represents different rates of evolution at different sites. And so these allow us to have the more complex models of evolution for the evolution of our DNA sequences. So how do we pick a model of evolution? Okay, how do we pick which one is best for measuring our differences and making that correction between observed differences and the actual distances, right? Well, what we do is we look at the actual data and we estimate based on that data what these different rates are. Maybe we have, and again, realize that this one going down the middle is really not very useful, but we estimate and we look at which of these fits best. So basically, we do a calculation to try and determine which of our models of evolution best fits our observed data. There is one additional thing that we need to consider, though, when we do this. Statistically, every time that we estimate a model of evolution, or sorry, let me step back. Every time we estimate a parameter, we add a level of statistical uncertainty to our analysis. And there's a trade-off there. Every time we estimate it, and we add this additional level of uncertainty, we lose our statistical power. We lose a little bit of statistical power. And so when we are determining whether or not we should use a more complex model versus a simpler model, there's a little bit of a calculation to determine whether that trade-off, the loss of power because of estimating additional parameters, whether it is worth doing, whether that trade-off is worth it. We do a little calculation. You're not going to learn how to do this. You don't need to do it. But we make a jump and determine whether or not that jump to the more complex model is worth the statistical trade-off. And then based on that calculation, we pick a model of evolution. Now, there's some other things that can be further complex, and we're not going to learn about these in terms of implementing them into our models of evolution, but just be aware of them. There are some cases where mutations may not be completely independent of one another. One very easy example of this is in ribosomes. Ribosomes form these stem and loops. You may be familiar with that already, and they're part of the three-dimensional structure of ribosomes that allow them to accomplish the purpose. So if I have a mutation at one position, it may very well have an impact at its partner site if it's part of one of these stems. And so we may not be able to treat every single mutation as completely independent of one another. Same thing might occur in proteins. There might be parts of proteins that fold over and interact with one another, and so mutations at one site may not be 100% independent of mutations at another site. And so this further complicates our models of evolution. So just be aware of this idea of independence and that we may not always be able to assume it. Be aware of these examples where independence may not be a valid assumption. Okay. Also, sometimes there are biases in base composition, and those might need to be included in our models of evolution. So in some bacteria, we have a biased GC complement, meaning there are more G's and C's in our genome than there are A's and T's. And so we may need to incorporate base composition into our models of evolution. We already talked about varying in rate substitution among sites, but that might get very complex in estimating our gamma distribution. It might be more complex than just simply picking which simple gamma curve is going to be the best. There might be more complex ways to estimate. So the idea is that we can get more and more and more complex models of evolution that better fit what's actually going on in the history of these sequences. However, remember, every level of complexity we add, we lose a little bit of statistical power. And so whether or not it's worth incorporating those more complex models is going to depend on whether or not that calculation works out. Now, a couple final points. Given enough time, sequences with higher rates of evolution are going to reach saturation high more quickly. However, sometimes there are constraints. We're not going to worry about what all those constraints are, but we might have different plateaus or different saturation levels for different types of DNA sequence. The ones that we are going to be dealing with most, however, are the ones that are unconstrained, that are just allowed to change and evolve and mutate without much constraint on them. And those are sequences that are evolving neutrally. Those are going to be the most important for us moving forward, and the ones that we are going to deal with. So for unconstrained sequences, I want to talk about where this saturation curve occurs. Okay? And so let's look at where that occurs very briefly here. And to illustrate this, I'm going to go back to our alignment here, but we're going to do a very simple one. So let's get rid of all of these. Let's add a brand new sequence. And again, to make it simple, let's just do 10. I think that's 10 there. And then let's imagine a sequence that diverges from this. And originally when it diverges, it's exactly the same. And then over time, mutations begin to accumulate in these sequences. We'll just add a couple mutations. and we would measure a genetic distance. Right? So now we have 3 out of 10, or a genetic distance of 30%. We had another one, and now we're at 0.4 or 40%. And so this is going to be this very familiar curve, right, where we, over time, accumulate distances. And so far, if this is a very consistent mutation rate, our genetic distance is going to be a good estimate of how much time has passed. But eventually, we're going to start accumulating mutations at the same position. So even though we know, because you watched me do it, that there are 1, 2, 3, 4, 5 mutations that occurred, if we came along and measured these sequences in these two species, we'd only count a genetic distance of 4. So we'd be missing one of the mutations. And so we're going to start reaching saturation here, and we're going to start seeing a drop-off from, let's do it this way. So here would be the actual genetic distance going from 0 to 100% difference at some point, right, where we'd be done. But in reality, what happens is we follow this line pretty evenly, but then we start to fall off as we reach this saturation curve. Okay, and so if I come in here and I change more things, add more mutations, and I just randomly do this, eventually we're going to reach a saturation point. And we're going to start having some of the mutations be covered up, some of the mutations be convergent, and eventually these sequences are going to become so different from one another that they're not going to be biologically useful anymore, right? And eventually they'll be completely different. Well, let's think about that. Are they really ever going to be completely different? Eventually, so many mutations are going to occur that these are going to essentially be random pieces of DNA with really no connection to one another at all, because so many mutations have occurred that we've covered up all of the historical similarities between them. However, in reality, DNA sequences never get to be 100% difference. And the reason for that is very simple, and you might have already jumped forward, or you might already know about why they never reach 100%. There are only 4 nucleotides possible. So as more and more mutations occur, as we essentially randomize the DNA sequence, eventually we will plateau out. And this plateau here is going to be at, that plateau rate is going to be at 0.75% genetic distance, or 75% difference. And the reason for that plateau is because there are only 4 base pairs. Once we have essentially randomized our DNA, we have a random 1 in 4 chance of being the same. So 75% genetic distance. So for unconstrained DNA sequences that are just allowed to mutate and change freely over time, eventually we will plateau out at 75%. If we have a higher mutation rate like this, we'll reach the same plateau level, but we'll reach it at a faster rate. So remember this initial slope here, which for this one is there, for the first one was right there. If we have a very low mutation rate, it might take us I'm going to go way out. It may take us a very long time, but we'll still plateau eventually at that same point. So, but that's for unconstrained DNA. For unconstrained DNA, we saturate at 0.75% or 75% difference. And that's because there are only 4 possibilities. Now, what do models of evolution have to do with this? Let's make this a little less busy. Models of evolution allow us to make corrections in this period of time. Before we reach complete saturation, excuse me, but when we are diverged from the ideal. And so a model of evolution allows us to make a correction over that time period. Once we reach into this saturation point, even the best model of evolution is no longer going to become useful because our DNA has essentially become randomized and there's no meaningful comparison between the sequences. Okay, but in the meantime, we can make those corrections, and that's why we have models of evolution. And that's why I'm asking you to learn those 5 basic models of evolution, plus the 2 additional parameters, the gamma rate and the proportion of invariable sites. Okay, so hopefully that makes some sense to you. Now, if we have other things going on, we could have different saturation points. If we have a constraint of evolution due to natural selection, or if we have second, first, and third positions of codon, we could have constrained DNA. But we're not going to worry so much about that. We're just going to worry about the unconstrained DNA, which is always going to saturate at 75%. Now, next discussion, we will talk about models of protein evolution and protein sequences. Okay? So we'll do that. We'll move forward. You have that to look forward to. And again, I know this is kind of a jump in and swim in the deep end lecture for many of you. I know that. If you're struggling, review it. Review the concepts. Look at the SLOs and see the SLOs. That will help you to simplify and understand some of it a little bit. And then if you're still struggling, log on to a Zoom session, make an appointment with me, and let's go over it again, or let's answer your specific questions.